you so much for the lovely introduction, and I'm very happy to be here. So yeah, that title is a bit of a mouthful, so let's break it down little by little, and let's start easy with SARS-CoV-2 evolution. And thank you so much for the brilliant introduction to this, because now this figure is almost self-explanatory. But so, like you said, there's these big gaps in terms of kind of like, so there may be a sequence, or rather a subtype that's floating around there, but you don't actually necessarily see it until you actually sequence it. And so because of that, you get these quite large gaps sometimes. And so the question is, well, so what's causing these gaps? So we can look back historically to the first time we saw a gap like that, which was when Omicron first came out. Yeah, when that happened, everybody was super scared because it all of a sudden had all of these mutations coming up, and we had no idea where they came from. So let's talk about why. So the question that I like to answer is kind of what can we do to slightly make these gaps a little bit smaller? And there's two ways to do that, basically. We can either get more informed with biology, or we can just simply throw more data at the problem. So the biology informed our patient selection. So basically, we wanted to see if you are immunocompromised, and that leads to typically longer infection cycles. If during that longer infection cycle, you can accumulate more mutations, and thus a single transmission event from such an immunocompromised host could cause these massive gaps. Or, which is what I'm going to talk a little bit more about today, it's my focus, which is the more data side. So before we really get into more data, we have to think about, well, how viruses are a little bit different from how we think about genome sequences in general, because every time they replicate, they have a complete random set of mutations that could be coming out of that. And well, what you get out is a quasi-species pool, where some of the progeny are identical to the original virus, but I'd say the vast majority of them aren't. And that pool then undergoes selection, and so it's quite interesting to know whether or not that pool is changing in response to treatment or in response to a whole load of other things. But so the way that we typically represent them, or how you're used to thinking about them, is through consensus sequences. And consensus sequences aren't great because it's kind of like taking limbs from different viruses and stitching it all together, and it's not very representative of any one virus in your sample, and it's also not very representative of the whole pool. So instead, what I'd like to have is kind of like a blended average of all the positions throughout the genome to get kind of a quasi-species informed sequence. But so what tool am I going to use to do that? Because there's kind of not really any tool out there on the market that could get me to this level of diversity. And that's going to be my focus for today. So you've all also heard plenty about multiple sequence alignment, but so kind of the main problem with it is that it can't deal with something that's kind of largely complex. So if you have something like this, which would be like a structural variant, then all of a sudden your multiple sequence alignment is failing, even though there is a kind of, rational evolutionary reason why these two things should be related. So as a tool, multiple sequence alignment is a little bit too coarse for the kind of problem that I'm trying to solve. But so we can do things a little differently. Namely, we can just look at the word counts because then, well, it turns out that they're identical, right? So if you're familiar with the field, this is kind of like looking at K-mers. So again, you can see very clearly that they're identical. And but if we wanted to take a look at something very different, and I'll tell you the genomic analogy for this in a second, they're going to come out to be quite different, even though, well, kind of the information in them is the same. And more importantly, the parts that are coming out identical are kind of the least important parts. So, you wouldn't necessarily care about them being the same. And so when you think about this in terms of genomics, this is a lot like convergent evolution. So you have the same function, but it's just written differently in the genome. So as a tool, this is also a bit non-specific. So again, we have to go for something different. And this is where we can leverage information theory. So we can pull out the most important parts of the sentence, the subject, the verbs that are being used, and then kind of fill it in with a whole bunch of extra information to get, a more informed setup. But so this is almost right, because now if you wanted to look back at, say, like a single nucleotide polymorphism, information theory, when you're thinking about these kinds of compressions, might not necessarily be the best tool for that. So let me tell you what is. So we have to think instead about representing genomes geometrically. So say, as it's going along its sequence, synthesizing it, sure, that could be one way of representing it, but it's not very helpful. So how about instead we ask the genome as it goes along its sequence to instead move in this kind of decision space where you're seeing kind of where it's going. And let's tell it, all right, so every time you make a decision about which next nucleotide to use, you have to go halfway to the next nucleotide. And so you get this kind of a visualization, and, it's going over and over. And effectively, it's an iterative function system because every following position depends on the previous one. And because of that, when you get this kind of final pool at the end, which looks very chaotic, it's completely unique. This representation is unique to exactly this sequence. And actually, if you had perfect mathematical and decimal precision, the last point alone would be enough to tell you your entire preceding sequence. But, well, obviously, computers don't work that well. So this is the kind of basis on which chaos game representation is built. For those of you who have been in the field for quite some time, you may be wondering, oh, why is she talking about something that was published in like the 80s or 90s? And it's really interesting, and there's a lot of new applications for it. But effectively, this is a visual representation, but it's just a list of coordinates. And these coordinates, well, you can use them just like alignment. Yeah, but so I told you I want to do a kind of diversity representation. So for that, we need to kind of manipulate our chaos game representations just a little bit. And here we can leverage something that's a bit special about them, which is that since you're playing it on this kind of like 2D Cartesian plane, the set of these the way that you can get into one of these quadrants is the only way, rather, that you can get into one of these quadrants is if the next nucleotide in your sequence is the one that's represented by that box. And that's not just for quadrants. You can actually divide this space again, and you can divide it again. And so you can get dinucleotide, trinucleotide frequencies, down to whatever you want. And some people actually even do fractional k-mers, but who knows what that means. And so kind of you keep doing this, and you can get this kind of a frequency chaos game representation object. But, you may be realizing that, well, this is effectively just a substring frequency table. And the special thing about it is just its layout. And it's because it arises through this kind of chaotic game that you play, and you get a very specific clustering of all these very similar k-mers ending up next to each other. And so how we can use this to represent metagenomes is, well, we can take our FCGRs of all of our quasi-species, and, well, they're just matrices. We can just take an average of every box in those matrices and get ourselves our metagenomic FCGR. And so you might be seeing that and have the obvious question, well, how did you even get those quasi-species FCGRs in the first place? And so now we get down to the nitty-gritty of my data, right? So we do short read sequencing, which means that we'll never be sure about which mutations happen with any others. So effectively, I don't care. I just oversample them and play a statistical game of chance to just do them all together. So like I said, we split everything into technical duplicates. We analyze every duplicate separately, and then we even go deeper because we want to go down to the lowest level of mutations that we can possibly get to. And to do that, we need to throw a lot of analytical pipelines at the problem. So we use 3 different pipelines in order to really be certain. So for every duplicate gets run on 3 pipelines. We then compile those 3 pipelines, and then we check for what mutations are present again in 2 duplicates. So there's really a lot of data curation that goes down so that we can be absolutely certain that every mutation that we have in our final table, down to 1%, we're certain that it's there. And then basically I'm going to make 10,000 CGRs, stick them all together, so that way I have a kind of a good representative of all the possible combinations. But so, okay, let's talk about how I'm actually going to make those 10,000. So effectively, say that we had these 2 mutations. So they're 50% frequencies for simplicity, but my program does actually do this in a frequency-dependent manner. So as you were to go along this genome, you reach the position where you have this frequency, or rather this mutation, except instead of the virus gambling about what mutation is going to happen, it's me gambling as I'm synthesizing this. But then it's not just that I'm only focusing on this one. Both of those then go on to create their own quasi-species, kind of secondary mutations. So that way you get every pairwise combination of every possible one following. And that way you can get kind of a very good representative pool of kind of everything's in its right frequencies, and you also get the full picture, right? So, okay, we've talked now about our encoding system. Now we have to think about our embedding system, how we're actually going to be manipulating these sequences. So all of this is based on this paper that's cited below. They're geniuses. I don't know how they came up with what they did. So I'm going to simplify it for you guys. Basically, you want to compare 2 FCGRs with each other. How you're going to do it is you're going to pretend that they're effectively just matrices. So what you can do is single value decomposition. You start by extracting your basis vectors or your columns. Then you apply what's in their paper, they call it a truncation dimension. And the way I think about this is you're just removing some columns that aren't necessarily contributing to the variability in your data set. So you're kind of like getting rid of the noise and the junk. So you're keeping the stuff that's actually really causing the variance. And then you compare the angles between all of those vectors in this higher dimensional space, and you get your summaries. And basically, you can just use the Euclidean distance to summarize all of those angles put together and get your one pairwise distance value. So, all right. I've told you a bit about my tool, and I've told you that it's great. So it's the Swiss Army knife of sequence analysis, in my opinion, because, well, you start with a CGR, which is effectively an alignment tool. Then you take that into an FCGR, which is a substring table, and then you can leverage manifold embedding to use information theory. So effectively, you've got all three types of sequence analysis in one tool, and you can kind of cherry pick what level you want to go to depending on what sort of question you want to answer. So, okay, now we can move on actually to the visualization step. But before we do that, I want to tell you guys actually what it's like to use my tool. So, okay, the first thing we have to do is decide how low we want to go in our mutation frequency. So, most people, when they look at sequences, they're mostly thinking either 99%, so full conversions, you have no ambiguity, or you can go down to 50% where you have some ambiguity and then you get those, fancy nucleotides like N coming up. But we care about really, really going down all the way to the bottom. So like I said, yeah, down to 1%. But once you've made that decision, then you have to decide the resolution of this object. So that's kind of how many boxes you're going to slice that space up into. And then you have to decide what's your truncation dimension. And in a microscope analogy, this is kind of like how much light you're actually going to be putting into the system to get the right kind of contrast. And hopefully, if you do all of those things right, then you can get out your evolutionary trajectory. So, okay, like I said, the thing that we had from our embedding were these pairwise distances. So let's take a look at this kind of heat map that we can get from these pairwise distances. So on the diagonal here, you get the darkest values, which are the samples compared to themselves, which obviously have a distance of 0, so they are darkest here. And you can start to see some trends when you start to think about inter-patients. So this is between patient comparisons. When you look at the groups, you can, treatment groups, sorry, and you don't have to necessarily know what treatments are which, but just to see that there is a general pattern that the samples that receive the same treatment end up seeming a little bit more similar to each other than they are with samples that underwent a different treatment. But if we go down to major variants, this looks quite different. All of a sudden, you don't have this blanket similarity anymore, and you start to see a little bit more clustering actually in the intra-patient level. So this is samples taken within the same patient just over time. And again, that's even better when you go down to minor variants. You can see that there's a very, very clear trend that shows that as you go down in, higher diversity down in mutation frequency, you can actually recapitulate way stronger intra-patient relationships. So that's great in terms of, leveraging the data that we already have. And this table just summarizes that, for you to be able to see it a little easier, that really going down to 1% is really beneficial. So, but what we can do from this kind of a representation is we can also apply multidimensional scaling because, well, you're not limited to looking at this as just a heat map. And this really, in my opinion, helps me illustrate the point that I'm trying to make. Because so when you're looking at the consensus sequences, sure, they're all very similar to each other, but the variation that you're seeing is very low dimensional, meaning that the changes that you see are very consistent and kind of everything's pretty flat. And then when you go and when you raise the diversity and you go to major variants, then all of a sudden you're starting to see this kind of higher dimensional activity, and things are happening kind of chaotically all over the genome. But very clearly you can see that there are intra-patient patterns. So you can see that there are these clusters forming. But yeah, when you go down to minor variants, you can see that they're really, really right on top of each other, especially these blue clusters, which are at the top here. You can see that those points are effectively just, on this same spot. So the work that we have still yet to do is to actually link this back to individual mutations. Like I said, we care about knowing whether or not the immune status or the specific treatments have an impact. And so there's no statistics on this slide because, like I said, it is very, very preliminary data. But the premise is that you can see that there are some trends. Yeah, so solid organ transplant individuals, which is the middle column here, those have induced immunosuppression because as you receive the transplant, you have to take drugs so that your body doesn't start fighting the transplant that you just received. Meanwhile, hematological is, the inborn errors. So you can see that actually kind of inducing the suppression actually might lead to you having more mutations coming up because your immune system just isn't as good as tamping down the infection. And when you look at the treatment patterns, it's not necessarily so clear, but that's because to really look at treatment, we have to look at the kinetics, right? We have to look at the trajectory. So we've made all of these figures to just look at the trajectory, but now it's the ongoing work is to link them back to the fractals, yes. But we can already see very clearly that the patient in this figure here, they received convalescent plasma, which for those of you who don't know, basically it was kind of our last ditch treatment effort when it came to when it came to treatment, you just took all the antibodies from someone who recovered, gave them to a sick person, and just hoped for the best. But so what that means is you can get a lot of new mutations coming up, and that's quite clearly confirmed in this sort of visual. So to conclude, using the quasi-species sequence, which kind of averages together all of these things, is the best way to go about it. If you really want to see these very close relationships, consensus sequences are still fine. It just depends on the kind of question that you're asking, and as well, doing what I did is quite computationally intensive. But so we can also leverage chaos in order to represent this diversity, which is how we made these metagenomic FCGRs in the first place. And if you use these, well, in the end, you get these really nice interpatient relationships. And yeah, paper coming soon. And I just wanted to thank my team at the LUMC. They've been incredibly helpful, and the names in bold, without them, this project wouldn't have happened. Thank you.
Thank you. It's a perfect time. So, any question? Here. Yeah.
So thank you very much. That was fantastic. So you say is it computationally expensive? How much and until what point you can reach? Like we are talking about like 6K. Can you reach 20? A million?
So the thing that's actually most challenging is the variant caller pipeline. So actually, when it comes to this manifold embedding and the fractals, that's instantaneous effectively. So actually running that, it's no problem.
Yeah,
like
matrices
seconds.
are great. Yeah.
But it's, sorry?
Matrices are great.
Yeah,
can
they
do
really
whatever.
are, absolutely. But yeah, but when it comes to the variant calling, one of these variant callers, Vardict, for some of our samples could run for over 30 days, so that can be quite prohibitive for some labs, yeah. But actually, I'm very, very glad you asked this, because another cool thing, and this is, kind of pocket information that I've got, that actually you can just use the raw reads and you get a comparable FCGR object.
Yeah, you don't need to map, I think.
Yeah, exactly. Yeah, you can just go straight from your data.
Yeah, I think so. Thank you. Fantastic again.
You have time, so more questions? No? Oh, here.
So just to follow up on this raw read idea that you brought out, does that require you to change sort of your minimum because of sequencing error, or could you expand just a little on the tweaks?
Yeah. So the reason that you can do it, and so they're not so immediately comparable to these kinds of, alignment-based methods where you're only including the specific variants, because you obviously overrepresent the beginning of the read compared to the end of it. But basically, you can use the chaos to kind of do error correction for you in a sense. Yeah. So when you already do all of this plotting from the raw reads and then you summarize it on this FCGR object, because of the kind of, there's only so much kind of frequency depth that you can have, especially if you consider it as an image. You only have 256 levels. If you leave it as a matrix, you can go as high as you want. But yeah, basically you can just use the chaos to kind of do error correction for you. And so there are people also who do Oxford Nanopore real-time sequencing. And so they use FCGRs to do this kind of real-time error correction for real-time data access. But that's a little bit more technical, because you have to kind of know what your data is beforehand, train this kind of convolutional neural network to do the, error correction for you. But yeah, that's kind of where I'm going to be going forward with it.
Okay. Oh, another one. So we have still one more question. So,
thanks. So the intra-host evolution that you talked about, is there any information on how that correlates with outcomes, with treatment outcomes?
Yeah.
Yeah,
so our patients are all from the ICU. So we only have one death in our cohort. So in the sense of, trying to do statistics on that, it's, a little iffy. Other than that, I don't necessarily have any quality of life information on these patients, unfortunately. The one thing that we are definitely doing is CT values, but, that's not necessarily such a good prognosis for, kind of patient outcomes. It's just what we can do. Good question. I'd have to ask my clinicians if they have that. Thank you. That's a good suggestion.
So,
thank you.